In 1952, Alfred Hershey and Martha Chase provided a confirmatory experiment to previous experimental results associating DNA as the hereditary material. They used T2 bacteriophage, a virus infecting E. coli cells, to determine whether DNA or proteins is the genetic material. Hershey was awarded the Nobel Prize in Physiology or Medicine in 1969. Chase was the laboratory assistant in the lab of Hershey. Unfortunately, she was not included in the Nobel Prize. They labeled the protein coat or capsid on one batch of a viral culture with radioactive sulfur or 35S, which is incorporated in amino acids, methionine, and cysteine. They also labeled another batch with radioactive phosphorus or 32P, which is incorporated into the viral DNA because DNA is rich in phosphorus. Then they infected E. coli cells with each batch separately. T2 bacteriophage has a lytic life cycle wherein after its attachment to the bacterial host cell, it will inject its DNA into the cell cytoplasm and use the host cell machinery, the ribosomes, to manufacture phage proteins such as capsid, tail fibers, and its DNA, which will later assemble to form new phage particles. After infection, they homogenize the culture using blender and centrifuge to separate the lighter viral particles from the bacterial cells. The heavier bacterial cell settled on the bottom of the tube, whereas the lighter phage particles remain suspended in the supernatant. They noticed that radioactivity remained only in the supernatant in the batch infected with 35S labeled phage. This suggests that the radioactivity remained only with the proteins in the phage particles and was not introduced into the bacterial cell. In contrast, tubes infected with phage particles with 32P labeled DNA, the radioactivity was detected in bacterial cells. This suggests that the radioactivity from the 32P labeled DNA in the phage particles has been introduced and expressed in the bacterial cells, producing new phage particles. This observation led them to conclude that the DNA, which was injected into the bacterial cell, carries the genetic material needed for the synthesis of new phage particles. Their result finally convinced the scientific community that DNA is the genetic material.